I brought my cells to the microscope and I found them. That's on the computer using regular light microscopy. And what I'm going to do now is I am going to turn off the white light. So there's no more white light coming. All fluorescent microscopes have something in common. They have a source for ultraviolet light to come to the microscope. So my next step would be to turn on my UV light box and increase my intensity of the light coming through. And so this is the back of the scope, and we can see this optic wire coming through, letting the UV light come to the microscope. So our next step is to let the UV light in by moving this panel so the blank spot is open and lets the light in. We'll be going over this in a stylized way, but right now we want to pick the appropriate filter cube with, this has a mirror that's going to move all the light. I have the appropriate filter cube established here, so I just pull out that stick. It could be a rotator. And the one important thing is I'm looking for green fluorescence, so I want to see blue. Okay, I turned out the overhead light. I want to see the blue. If I was looking for red fluorescence, I would want to see green. So this is being excited by green light, and I'll be able to see red. This is being excited by a blue light. I'll be able to see green. This is being excited by ultraviolet light, which we cannot see, and then I'll be able to see blue. The phalloidin is with 488, so I want a blue light. So we're having our cells exposed, excited with this particular light, and we are looking over at the cells underneath the scope, and they are fluorescing green. Now a nifty trick we can do is we can turn on the white light again, and what that'll allow us to see is the outlines of the cell, and then I'll be able to dial that back and then see the fluorescence come back. And then this is what I see with plate number 1. There is no fluorescence. But if I toggle on the white light and I turn up the white light, I'll notice that there are cells. They're just not fluorescing because they didn't get Triton, and the same for the plate that didn't get phalloidin. So if they didn't get Triton or they didn't get phalloidin in the presence of Triton, they don't glow green. If we want to take a picture of this, we just have to take a snapshot. So I'm going to take a snapshot, and then we'll look at particular ones when we get together. Snapshot. Okay.